Okay, good afternoon. Welcome to the first panel discussion of the afternoon at the ARM Cell and Gene Investor Day. I hope you had a good morning and a nice lunch. The Immuno-Oncology Pipeline for Liquid and Solid Tumors is the title of this panel discussion. And what we'll be speaking about is that in the wake of last year's approvals for ALL and for DLBCL, we're all eager to understand the timeline and what has to happen now for new clinical breakthroughs in liquid and solid tumor treatments, including promising data for multiple myeloma and other indications. We've assembled a panel today that's uniquely qualified to speak about this topic, and I'd like to get started by just putting a little bit of context and framing around the session. The major therapeutic platforms and enabling technologies that exist today, specifically related to the IO dealing with the IO pipeline, are advanced cells, modified T cells, of which you've seen a lot today and will continue to see, cell-based immunotherapies, including T cells, CAR Ts, TCRs, NK cells, TILs, MILs, marrow-infiltrating lymphocytes, gamma delta cells, and dendritic vaccines, etc. And then novel and synthetic gene delivery vehicles, including adenoviral-associated vectors, lenti, retro, and adenoviruses. And then moving into viral and non-viral-based gene editing technologies using CRISPR-Cas and next-generation CRISPR-Cas technologies, TALEN, zinc fingers, and homologous recombination and the like. And then following behind that, next-gen expression constructs, including novel capsids and innovative regulatory elements, which can enable specificity, strength, and improvement of capacity, plus inducible elements to regulate either temporarily or in response to external stimuli for purposes such as molecular kill switches to improve safety of these products. And as you can see here, there are a number of companies and research institutions that are focusing their work in these areas, in the CAR T space, in the space of TCRs and modified T cells, gamma delta T cells, NK, and once again, tumor-infiltrating lymphocytes and marrow-infiltrating lymphocytes. So without further ado, I'd like to turn over to the panel and have the panel to begin introduce themselves, what is their, who they are, what their role is, what their company is doing, and give you a little bit of sense of why you should be listening to them today as they discuss these important topics. We'd also please encourage you, if you have any questions, just to raise your hand, and we have a microphone, and we'll take questions from the floor. Thank you. Samuele.
Yeah, so good afternoon, and thank you for joining this panel. Thank you for the co-panelists. My name is Samuele Butera. I lead the cell and gene efforts globally for Novartis, and we are very proud to be on stage here with these other companies and the companies represented in the room that are really pushing the boundaries of what is possible for patients worldwide. As the audience is aware, we were the first one to launch a CAR T in the US in pediatric acute lymphoblastic leukemia in August of 2017. We are now up and running, more than 6 months after, with patients benefiting from Kymriah. We're also getting ready to launch diffuse large B-cell lymphoma in the US, as well as both pediatric and diffuse large B-cell lymphoma in Europe. We should be hearing from the regulatory authorities anytime, and also expanding beyond those geographies into other parts of the world where we conducted clinical trials. As we go through, I'm sure the next 45 minutes, we can talk more in terms of the overall commitment that Novartis has in cell and gene therapies, which is not just in CAR T or in autologous therapies, but also in allogeneic therapies, as well as not just in oncology, but also in ophthalmology and other areas, as you've seen recently from some of the BD&L activity that we've been performing. So look forward to the panel, and thank you all for joining.
Thank you, Samuele. Jennifer?
Okay, thank you, and thank you again for having me on the panel as well. My name is Jennifer Butler, and I'm the Chief Commercial Officer and Head of US for Tessa Therapeutics. So Tessa is a private global biotechnology company that's focused on T-cell therapy, and specifically virus-specific T-cell therapy. Right now we have an ongoing phase 3 program with a target of 330 patients in 5 countries with 30 sites. We actually deliver our cell therapy out of a central manufacturing facility that's based in Asia. And so our vision and hope is that we can actually bring these cell therapies to Southeast Asia and Asia, as well as the US. We have an ongoing trial here in the States as well. So we're looking to take this platform. We think there's great utility for T cells, and specifically T cells that target virus antigens, virus-specific T cells. So obviously with diseases like nasopharyngeal carcinoma, which is EBV positive cancers, as well as things like cervical cancer and head and neck with HPV positive, as well as really looking at novel combinations to kind of bring forth this idea of taking tumors and putting viral antigens on the surfaces and then redirecting virus-specific T cells to them. So that's just a little bit of a background on Tessa Therapeutics, and looking forward to be part of our discussion today. Thank you.
Yeah.
Good afternoon. Derrell Porter, Head of Commercial for Atara Bio, which is a San Francisco-based publicly traded company. We focus on off-the-shelf T-cell immunotherapy. We cover 3 different areas: oncology, and has both solid tumors and hematology. Also viral diseases and multiple sclerosis, so CNS as well. And we have a late-stage program. It's called tabelecleucel, and tab-cel for short, given that that's a mouthful. So we just initiated 2 phase 3 trials in EBV positive PTLD, which is post-transplant lymphoproliferative disorder. Obviously, we call it PTLD for short. So we've initiated 2 phase 3 programs, both in the stem cell transplant setting as well as the solid organ setting. And we expect to file a CMA by the first half of next year with plans of commercializing in Europe first and then subsequent to that in the US. We also have a program that is very similar to my colleague here. tab-cel is also pursuing nasopharyngeal carcinoma in combination with Keytruda. We're also looking at a number of different viral diseases, including CMV and JC and BK virus. And so our program is really focused on off-the-shelf, and I think that'll be an interesting topic as we discuss here the difference between auto and allogeneic, and we really believe in the benefit of an off-the-shelf approach, really getting our therapeutics to patients as soon as possible.
Thanks, Derrell.
Jeff Walsh. I'm the Chief Financial and Strategy Officer for Bluebird Bio. We're a company that has really 2 different approaches within the gene and cell therapy. One is focused on targeting hematopoietic stem cells for severe genetic diseases. And then we also have a separate platform that is targeting T cells, obviously relevant for this discussion, in oncology. And speaking of that platform, we have an ongoing, with our partner Celgene, program in multiple myeloma called the KarMMa study. We've disclosed data over the last year and a half on the phase 1 that led to the initiation of the KarMMa study in multiple myeloma. That's a CAR-T program that is hopefully marching toward a filing next year. At least that's the current expectation. And the intent of this year also to start a trial in third-line therapy. The KarMMa trial is in relapse refractory patients in multiple myeloma, so the very sick end of the spectrum. Behind that, we have a series of both technologies, products, and partnerships that span from TCRs to CAR-T to gamma delta T cells through both internal programs as well as partnerships with the likes of TC Bio and Metagene. We believe some of the leaders in their respective fields. And we also have technology that we've accumulated over the years to apply to the solid tumor environment. In particular, we have a gene editing platform that we think is going to help us in the solid tumor environment. And we've disclosed some data recently on the application of that gene editing platform, at least preclinically, and most recently at AACR. And we can certainly talk about why we think that has application. but it really does span the ability to target solid tumors, liquid tumors, and even some of the off-the-shelf approaches with gamma delta T cells, where we think that has significant merit for that approach.
All right, thanks, Jeff. So as we evaluate the cell and gene landscape today, and we look at what the CAR T cells have done, particularly as they've gotten everybody quite excited, we've been able to break through the approval barriers in significant and meaningful ways. That begs the question, two questions really. One is, how's it going? And maybe Samuele, you can help us with that one. How's it going once we've broken through? And the other question will be, what's next? Where do we go from here? So maybe we can start with you, Samuele, and just tell us a little bit how is it going at Novartis now that you've got Chimera out there and the gates are open.
Gates are open, and we are very satisfied with what we're seeing in terms of patients, pediatric ALL patients that didn't really have any other chance or opportunity to be able to access, a therapy like Kymriah. We're also very excited to see, obviously, the opportunities in diffuse large B-cell lymphoma. We've had, multiple patients on therapy. We have multiple centers open, more than 35 at this stage, are able to administer safely Kymriah in the US. And as I mentioned before, we are also getting ready for a much larger launch, which is diffuse large B-cell lymphoma, where we hope to access a population that is going to be, orders of magnitude larger than pediatric. And again, offering an opportunity for patients that really do not have any other, viable option at this stage. So that's really kind of how it's going in the short term. And as you've seen from the press, we've been, I would say, careful and progressive in the way that we've expanded access to the different centers. So we want to make sure that, again, this being a very crucially new therapy, that the centers that administer this therapy do it in the most safe and effective way. We're going to continue that approach also for diffuse large B-cell lymphoma and as we expand internationally. But as you hinted, there's much more coming in the back. And Novartis, as you've seen across from media outlets and IR relations, et cetera, is really investing heavily in cell and gene therapies. And I would say there's probably three layers that we can talk about. The first one is the most comprehensive layer, which is in which therapeutic areas are we playing. And clearly we have a very heavy focus in oncology, but we are also having partnerships in ophthalmology, as I mentioned before, with Luxturna on the ex-US front, as well as the Avexis transaction that is currently underway. So at the highest level, Novartis is really committing to cell and gene, overall, regardless of the therapeutic area. Now, if you dive deeper and you think about oncology, there's two major buckets that I would talk about. One is autologous therapy, which is where we are all playing today. And then there is allogeneic therapy, which is a little further out. Within autologous therapies, we have four major avenues of, I would say, development and progress. Number one is geographic expansion with our current indications. So we are in the US today. We're going to expand in the US hopefully shortly with diffuse large B-cell lymphoma. We're going to go into Europe, and then with both indications. And then hopefully shortly thereafter, we're going to go into the other geographies where we had clinical trials. So that's the first axis. The second axis is around expanding the indications set with our current CAR T platform. And that goes both in existing indications with pediatric and diffuse large B-cell lymphoma, where we go towards the very high risk very high risk population in pediatric, and diffuse large B-cell lymphomas after first relapse. So that would be for the first two. But then we're also assessing with our partners in the University of Pennsylvania and other collaborations, how are we going to play in follicular, how are we going to play in multiple myeloma with using a BCMA platform, et cetera, and many, many other indications behind that. So that's the second axis, which is expanding indications. The third one, which might come up in the panel shortly, is how are we going to do, we've shown success, and still a lot needs to be done, of course, but shown success in hematological malignancies. How about solid tumors? So we have a third axis that is focused on solid tumors. And there, what is likely to happen is not just CAR T, but obviously some level of combinations with other agents. And then finally, and this is something that is very exciting with our collaboration with the Novartis Institute for Biomedical Research and other partners, is what are the type of improvements that we can make? And so, one improvement could be around, for instance, combination therapies with IO, right? In certain particular indications where we're seeing some great efficacy, some great safety, but something more could be done. So that's one category. Another one is around multi-targeting. So not just going after one, say CD19, but also other targets. And how are you going to do that? Are you going to do into a dual construct, tandem constructs, whatever it may be? How are we going to leverage that? And then thirdly, and crucially also, how are we going to innovate not just from a molecular perspective, but also from a customer model perspective, and really make CAR Ts, again, autologous CAR Ts, even more accessible overall. And then finally, I said that the last bucket would be the allogeneic platform, and there our partnership with Intellia and their CRISPR-Cas9 gene editing platform is something we're working very heavily on, and we'd love to see results, obviously, in the medium term.
That's quite an ambitious agenda.
We're trying.
Yeah. So it's fascinating. I mean, you covered off on solid tumor question, which I think we'd like to talk about on other ways to edit the cells and to get the genes in. You've covered off on geographic expansion. We talk about the delivery of particularly patient-specific cell therapies, which many people believed could not be done because of the quality, cost, scalability, and sustainability formula. You've proven now that this can be done. It's being also done by Gilead Kite. It's being done on large scale. So we know we can do that now. Let's talk a little bit about the next, how we can make improvements to that, and we'll take that down the line a little bit. Maybe Jennifer, can you talk a little bit about your strategy and what are we learning from this experience at Novartis now that we can take to the next step?
Obviously, we're all watching intently to see that. Obviously, there's a lot of barriers that are broken down, even from the standpoint of the fact that the institutions, right, where some of these cell therapies are delivered by the transplant team, and now you're trying to get the oncology team. And there's a lot of coordination that we're going to see. So I imagine that there's a huge piece that gets done just organizing the institutions around being able to deliver this. Particularly for, I think, Tessa, just because of the origin story of really making a decision to make the geography piece that you're talking about, to actually centrally manufacture that out of Southeast Asia. The idea was to deliver the cost basis inside the market that you might be delivering. So that was kind of something that we were able to do. And so when you talk about being able to commercialize a model, maybe in non-US or even Europe, I know others will go to Europe before we do that. But that's been kind of a way to potentially address that. And so being able to successfully kind of, deep freeze the cells and ship them to the 5 different countries has been our model. Now, we're still clinical, so then we're going to have to obviously, determine, how we're going to make that work commercially. But that's just kind of one example of being able to in some ways, sometimes the way the cells are produced and the way we can actually distribute them enables a certain model. But we'll have a situation because we are multiple doses. So all of these products have their own unique characteristics that you have to manage when you go in a commercial marketplace. So we have a multi-dose. So how do you essentially manufacture, ship it into a country, and then multi-dose, which might be obviously not the thing that you're managing now, but many of us would be managing in the future.
It's interesting that you say that because we've watched for years business models being built around manufacturing weaknesses or strengths, right? Just basically adopting the business model for a particular product. That level of complexity, though, is difficult to manage long term. And so maybe as we move beyond that and we move into the allogeneic space or another space, Derrell, maybe you could talk a little bit about your approach.
Sure.
And moving away from the autologous space more into the allogeneic?
Oh, absolutely. So we anticipate that we will be the first approved allogeneic product of this class of new therapeutics. So Juno would probably be next, and then Atara. And the way our system works and the delivery and administration of Tab-cel is that because it's off the shelf, we basically HLA match the patient to a specific cell line. And then once we receive that information, then we ship the product to the patient wherever they are. And with the expectation that they would be able to receive the therapy within 3 to 5 days, hopefully less than 3. And at this point in time, our benefit risk profile is relatively well balanced in the sense that it does not require any lymphodepleting chemo, obviously. Given the nature of the product, number one, and the side effect profile is relatively clean. So you just need 2 hours monitoring after administration of the product to make sure the patient's doing well, and then they're free to go back to wherever they are. And so we think that this does represent the next step of evolution in terms of delivering cell therapy, but then also just the administration and monitoring around these products. Obviously, we publicly disclose both some phase 2 data as well as our early access program data across a number of centers at ASH. And so the efficacy data is really, really compelling right now for tab-cel. And we look to continue to push the envelope, not only to take tab-cel to more indications. So the first one I mentioned is PTLD, which is a lymphoma, but we're also targeting a solid tumor with NPC and possibly other lymphomas with tab-cel. We expect to take it beyond that and actually broaden our overall portfolio, and 621 is a program that's targeting BK and JC virus, 2 viruses and 1 therapeutic. And then obviously we have an MS portfolio with 2 programs, ATA 188 and 190. One's off the shelf and one's an auto program. And ultimately that would be the first program, at least to our knowledge, that's targeting multiple sclerosis with a T-cell therapy. And so, again, I mean, we're watching Novartis, we're watching Kymriah, we're watching Tessa, obviously, and monitoring what all of our competitors are doing, as well as just the innovators in the space to see what we can learn from them, both from a scientific and clinical perspective, but also commercially. And on the commercial side, I think I'll just close with the point, the tables are turned. We do expect, and it's a great opportunity for Atara, that we will be the first off-the-shelf company and product that will be approved, hopefully, and therefore all of the commercial requirements and distribution and supply chain requirements to get the product to patients, will be leading the charge. One of the things that we've developed and we publicly disclosed is our product delivery system. It's called Atara MatchMe, and this is a system that really enables the receipt of information about the patient, so we get all the relevant data about a patient. Once we make the cell line choice, then we get the product to the patient, and that's all enabled by the system.
Maybe you could say a little bit more about that. I mean, that's it. I was going to ask you the question. How are you getting around the little problem of HLA, which has been around for a while?
So we have, what we receive is a high-resolution HLA data, for the patient at interest. And we have a cell selection algorithm that was initially developed at Memorial Sloan Kettering, and we've continued to refine that algorithm over time. And so using the patient information and running it through our proprietary algorithm, we actually choose the appropriate cell line that's appropriate for that patient. And then we ship the required doses to the site of care.
Hey, Jeff, are you thinking allogeneic will replace all autologous therapies someday? Is there a place for everything?
Well, I think there's a place for everything, and these technologies will evolve over time, and we'll hit our snags collectively as we progress, whether it be on the science side, the clinical side, or the commercial side. So I think we have to expect that is going to happen. We faced it ourselves in our sickle cell program as an example, where we thought we knew what was going to happen clinically, because we had one patient that was reported in New England Journal of Medicine, and suddenly we realized we didn't know really what we were doing. I mean, that's maybe an exaggeration, but we had to go back to the drawing board to really understand the biology of sickle cell and the protocols and manufacturing process that we were leveraging to see if we could solve the new revelations on the biology side. It took us 2 years to get there. We're hopefully there now. The data we disclosed recently at ASH suggests that we're heading in that direction, but we need more data. But that's just an example of we're going to face our bumps collectively across the board. And but I think there's a role for all. I think that's kind of our approach at Bluebird. We too have a bunch of horizons that we're looking at. Certainly with our multiple myeloma program, it's all about getting the pivotal trial done through the regulatory body and launched, assuming the data continue to look promising. But then it's earlier lines, third line, hopefully starting this year, at least that's the plan, and then potentially moving into second and maybe first line in the high-risk patients. And that's certainly some conversations that we're having and have been having for quite a while with Celgene based on the data. And then there's the considerations, okay, that's in multiple myeloma with our lead program. But to your point, what's next? And for us, we look at it based on the challenges. So they're fairly obvious. You've got antigens. How do you find more antigens like CD19 and BCMA? And so we've embarked on a pretty rigorous effort to find players outside of Bluebird that we think can find antigens for us, some of which are disclosed, some of which are not. But metagene's a perfect example of that, TCRs. So that antigen's a big part. Then there's a tumor microenvironment. And that's where a gene editing platform has a big potential role there. And we have some data, again, I referenced at AACR, that showed using a gene editing tool, you can, preclinical data, you can knock down PD-1 at the same time you express IL-12. So you, in the same construct, are able to suppress or inhibit the suppressive elements of a cancer cell and activate an inflammatory response locally on those cells. So we think this is the future, and we're starting to see that data. Others are obviously starting to publish on these type of things. So that's really important, and just one example of how we're approaching it with our MegaTau platform. So antigens, obviously the tumor microenvironment. And then beyond that, how can we apply this even more broadly as we learn more and more about T cells? Autoimmune disease is certainly one that we've been thinking about for a while and have some ongoing skunk work projects to think about that really next horizon. And so we've tried to collect's the wrong word, but acquire or build or generate tools that we think are going to tackle the key challenges that we inevitably as a field are going to face going forward. So that's the way we approach it. And I think the innovation on the commercial side is really relevant. And some of the stuff that you guys are doing is fantastic. And we are all learning from that. And it's kudos to you guys for taking some pretty big steps. I think we got to take more steps, and I'm sure you guys are working on that as well. That's where some of the learnings from launching in Europe for our programs, hopefully, in transfusion-dependent thalassemia and CALD, will give us some early learnings that we can then apply, like you're probably applying, to the commercial launch with Celgene in the U.S., and then obviously beyond that. And there's lots of challenges there, for sure, that you guys are living through right now, or we're just really starting to learn about as we prepare, hopefully, for a launch of TDT after filing this year.
One of the changes that's happened over the years, particularly in the patient-specific cell area, around the cell therapy space, we saw very, very little toxicity early on, but we also saw moderate therapeutic effect. As we're seeing more potent products now, we're seeing additional amount of toxicity, and I know toxicity is on people's minds. Can you guys talk a little bit about your view on the toxicity of the therapies that we're dealing with, this group of therapies? How different is that than small molecules, antibodies, traditional pharmaceuticals, and how are we moving forward to manage that?
Sure. Maybe I'll just take that. And maybe building on, Jeff, you talked about antigen, and you talked about some of the tumor microenvironment. I think specifically because of our platform really focusing on a T cell and a very specific T cell, the virus-specific T cell, I think, what we're also, I think, hearing about is also about the T cell T cell, and really focusing on what is the right T cell, what is the actual cytokine release profile that you want to have, and sort of that you could potentially moderate some of those having kind of all T cell activated. So for an approach, for example, with Tessa would be to focus on getting just that antiviral response versus potentially generating a whole bunch of other type of T cells being activated with that kind of on switch with the CAR. So I think that will also be an evolution. So not only finding those targets of interest and also kind of looking at that, I think that's going to be potentially where you're going to see some of these advancements. So I think it's going to be this constellation of looking at different antigens, and like I said, from our standpoint, actually looking at the T cell and what's potentially the best.
How big
T
a
cell.
problem is the toxicity today, first generation?
Well, I don't want to. Maybe I would probably want them to comment on it.
Yeah, sure. Come on.
So I think, obviously we take toxicities very seriously because, as I mentioned before, our main objective is to ensure that the patient receives Kymriah in a safe manner, and that is managed through the process that goes pre-infusion, during infusion, and post-infusion. And the two most important toxicities that we see are cytokine release syndrome, which is obviously a common toxicity in CAR Ts, as well as neurotoxicities. Now, we have not seen any case of cerebral edema in our own product, but obviously we keep monitoring for these things. And what we find out is really majorly three things, I would say. The first one is that when you talk to ultimately the physicians that are administering these products, they typically are transplanters or on-call transplanters. And while CRS and obviously neurotoxicity are something to take very seriously, they have been dealing with very serious conditions for the patients that have been on transplant. So it's not entirely unexpected to begin with. But then the important piece, and that's the second consideration, is that there is a pretty steep learning curve. Like if you talk to, for instance, Steve Grupp, who was our lead investigator on the pediatric trial for Kymriah, he's literally the management of cytokine release storm and the algorithm that we use has evolved significantly, as we gain more and more information about how to best treat those patients. And so now, whereas, for your first patient and you're a new center and you've never done, CAR T before, you tend to have this first patient in inpatient setting and you're, obviously following a very rigorous, procedure, et cetera. As you gain experience, the ability to manage cytokine release storm in a very effective and predictable way seems to be increasing with experience. And same thing with neurotoxicity. So the third consideration is then that, we as a company and all of us as an industry, investing in ensuring that we continue to address and evolve how the algorithms evolve, because the algorithm for Kymriah is different from the algorithm from other products. And it is as important to understand your own product as it is to understand that, on the other side, you have a center that at one point might have to deal with, a product from Novartis, a product from company X, company Y. And so how do you make an algorithm that is, effective for the patient first and foremost, but then doesn't confuse, the center too much, because it's going to have to deal with a lot of different constructs.
You have an answer for that?
We have the company, our own answer in terms of how we're building, our algorithms. We have been having conversations with other elements of the industry to see what can be harmonized, and there's definitely some common ground that can be found. But at least where we are with the technology today, I would say that there's always going to be a certain component of the algorithm that will have to be, product specific.
Maybe I'll add one more comment to that, just relatively short. I think it's also patient population dependent. The answer to that is if you're in, for example, the population that we show data on, the relapse refractory multiple myeloma population with an average of 7 prior lines of therapy that they've been exposed to, and you have compelling clinical outcomes from those patients, relative to standard of care in that patient population, you can tolerate a bit more toxicity, obviously. As you think about, and we've been fortunate enough, at least data we've shown, to actually have a relative to other CAR Ts, a pretty reasonably safe and manageable profile. But as you move into earlier and earlier lines of therapy, obviously that challenge becomes, that risk-reward question physicians are asking themselves, it gets tighter. And so that's as we collectively with Celgene start thinking about third line, second line, first line, we're having to look at the data and ask ourselves, do we think that therapeutic window warrants it relative to standard of care? So I think that if we want the CAR Ts and allo and whatever, TCRs and the like that are going to merge, to be broadly applied, this is the challenge that we're going to face going forward in terms of expanding within indications.
So I think we'd be remiss if we didn't talk a little bit about what's next on the therapeutic horizon. So we all feel, I think, multiple myeloma almost feels like by extension that sort of liquid tumor will be taken care of. So that maybe that's overly optimistic, maybe not, but for me, who's not involved in it directly, it's easy to say. So I see a different view. Let's talk about solid tumors a little bit. What's the problem Why can't we just do solid tumors, and what are we doing to address that?
I mean, I think Jeff touched on a little bit with just like given the tumor microenvironment specifically, the physical barrier and those type of limitations. I know one approach that we've taken at Tessa for kind of, I'll call it our next generation T cells, is to look at doing gene modifications to them. So one specific one is TGFbeta. So it's becoming pretty well characterized out there in the scientific community that you want to be able to basically have your T cells be able to persist in these kind of very high kind of TGF highly immunosuppressive environments. So I think what you'll begin to see is as we begin to understand what is it that we're kind of fighting when you get into the solid tumors, we'll be able to create therapies that persist, penetrate, and are able to generate that clinical component, unlike the hematological malignancies that we're challenged with. So I think everyone probably has an approach, but that just is one example with like the tumor microenvironment for us.
And I would add combination therapy, I think, is the other approach. So obviously in MPC, we're combining with an IO, Merck's Keytruda. So I think not only combination therapy, but also, I think Jeff touched on it, just with some genetic engineering, trying to attack this issue about the tumor microenvironment. So I think that's probably the next generation across the board.
Maybe the only other thing to add would be, on top of these two dimensions, is finding those targets that maybe are most receptive to, say a CAR T construct plus something else in that particular solid environment. So for instance, EGFR or mesothelin, these type of areas where we think that CAR T might be more effective in the short term.
So what does that do? TCRs, neoantigens, all these new targets, what does that do to the complexity that we're facing now? Does it help us, or does it give us more to do?
I mean, hopefully not. I mean, I think, who knows, right? We'll see what happens in the future. But I think a lot of the challenges that we've highlighted, I think Samuele's point and Jeff's is spot on. I think it depends on the tumor type, number one. Number two, I think that as the overall industry, and particularly the physicians and healthcare providers, get more used to using these products, we'll develop just pragmatic practices, frankly, in dealing with both the administration challenges, but also the side effect profile. So I think it depends, right? And we'll see what happens. But I don't anticipate any, hopefully, new challenges that will emerge once we start to develop therapeutics for solid tumors. At least on the horizon, it doesn't look like there's anything novel that's going to be a big challenge.
Okay. And I would say that we don't yet know in solid tumors what we're facing with standard therapies. So I think there's a lot to learn going forward, not just in some of these new targets, whether it be TCRs, neoantigens, or the next wave of target identification. There's just so much still to learn. We're still in the infancy of this field. And what we know is not a lot relative to what we're going to know in 5 years.
Completely agree.
And we hope that there won't be new challenges introduced by virtue of these new antigens, but yeah, we don't know until we get into the clinic and really start experimenting more broadly. Obviously, preclinically first, and then hopefully some things will come out of this that we feel good about putting into the clinic. And I know there are already some active programs that are showing some promise, so that's a good sign.
Well, it's certainly helpful that again, over the past couple of years, the business models that have been put together, the companies that are partnering with other companies, and the experience that's coming into the industry is helping us deal a lot with this complexity issue. It's not a bunch of small little companies anymore trying to figure this out. We've got a lot of support. So,
yeah, although we're leveraging the expertise of these small little companies to educate us, so there's still a lot of that. And these are smaller companies that may end up becoming pretty compelling big companies over a period of time if their technologies work, and that's the excitement of the field. You see a lot of these companies popping up that have these novel technologies, and now there's a lot of players to fund it going forward. That's the fun of it.
And all learning from each other.
Yeah, all learning from each other.
And I mean, the other piece I think, and Jeff kind of hinted at, is just the speed at which innovation is taking place. I think Usman Azam mentioned in one of the side sessions before, but if you think about example of Kymriah CTL019, we went from first in human to approval in 5 years. Right. and similarly for other players. And I mean, I think it's a great time of hope for patients. And the challenge will be, initially we're probably going to have, it's better to have many options and then see which ones work than having no options and being left without it. So it's going to be some adjustment period, and then the next wave is going to come up. And but I'm optimistic.
At this point, let me turn to the audience. Are there any questions people would like to ask of the panelists? Or should we just keep talking? Janet?
Experience with the clinical sites,
Yeah.
The dynamic between, you talked about somewhat the transplant folks
Sure.
and others, the desirability, what the demand is to become a site given some of the issues with the DRG at this point.
Sure.
Sort of that whole kettle of fish.
Yeah, no, I'd be happy to, and I will not go in too much detail because our own Q1, investors call is on Thursday, so obviously there's not some things I cannot share. But at a high level, I would say the first important, crucial difference between, I would say, what our industry, and through your help as well, is doing versus, small molecules or biologics is that in order to have patients have access to this therapy, there has to be unprecedented levels of collaboration across functions within the company and then at the hospital. And I'll exemplify it maybe in a couple of ways. One, 3 major layers of the organization internally are coming together. One is development. Second is, broadly speaking, manufacturing and supply chain, because you're dealing with an autologous therapy. Some of these therapies are cryopreserved, like ours, both before and after, they go to our own our manufacturing center. Others are not, but it's very, very unusual. And it's singular, it's personalized therapy, so we have to make sure that it goes to exactly the right patient. And then commercially, both in terms of access as well as support for the site as we educate the site. So these 3 kind of functions that typically lived in silos, definitely in small molecule, to a large extent in biologics, have to come together compliantly in cell and gene therapies. So that's one piece. The other piece is, who are you dealing with their centers of excellence? And I would say there's 2 layers here as well. The first layer is, obviously you want to make sure that all the key stakeholders are educated properly at the center of excellence. So that does not mean, again, like small molecules or biologics, that, okay, you're dealing with the physician and maybe, a couple of nurses, et cetera. No, you're really dealing with the entire, flow of the hospital, from the, administrator, figuring out the access and the reimbursement, all the way through the transplanter, to the nurse, to the ICU, because if you provide steroids, then maybe you actually kill the T cells that you're actually trying to grow. So all of that requires a infrastructure from the company perspective of very qualified individuals that are even more senior and even more qualified than your traditional commercial infrastructure. And then as important is also the surround effect that you have. In other words, because of the pioneering element of this therapy, there's not going to be hundreds of thousands of centers that are going to inject a CAR T. So what you have to do is ensuring that you have an appropriate referral pathway from the community hospitals and community physicians to the centers of excellence that can administer this type of products safely. And so companies like ourselves that have the infrastructure to have that proper education, both from a on-label commercial perspective as well as from a medical affairs perspective, that type of education has to take place, because otherwise the patient that could benefit from this therapy, particularly adults, because for pediatric, those networks are very well established, typically, pretty much in every developed economy. But for adult patients that maybe don't want to travel as much, maybe tend to stay a little more in the community, if we don't educate them properly, those patients are going to get stuck there and maybe die because they don't get to the solution that could be good for them.
Yeah, maybe add one. I'm sorry, Derrell. Just one real quick thing to add to that. Just on the front end of that, it's all about capacity.
Yes.
If we aren't able to scale this to a level that allows all of this intricate involvement to take place, then we will fail the patients. And I know we've been working with Bob's company for a number of years now, and I know you have a deep appreciation for how to scale these things. And we're spending a lot of time on figuring that out first and foremost to make sure that we can supply the markets, whatever the markets are. Sorry, Derrell.
No, I was just going to piggyback on the first question to you, Samuele. Can you share with the group one unanticipated surprise? I'm sure you had many, but just one particular surprise that you guys, there was a lot of planning that went into the launch.
Sure.
I'm sure you hit some things that you guys didn't anticipate.
Oh, look, 4 seconds left.
Whoops.
Wait a second, I want to know the answer too.
We are a learning organization. So. But definitely, look, you have lessons every day, and actually that is the beauty, in a way, of the structures that I think overall all of us are putting in place. And I would put it back to the importance of the integration, cross-functional integration, because a lesson that you may learn from somebody in supply chain has to be relayed very quickly to the account manager on the field, and maybe go back to the development guy to say, hey, look, we observed this, it resulted in this, let's change it there. And so you cannot think in terms of, okay, this is a stationary thing that is going to be like this for the next 15 years. Every day we learn something new, and actually, in a way, we were lucky to start with pediatric that has a smaller patient population size so that we could do things safely and appropriately, but at the same time learning because we had the time. And I would say less of the pressure than when you have thousands of patients all at the same time.
Okay, thank you. We are officially out of time. Please thank the speakers. Thank you.
Thanks, Andy.